Hello, everybody. So we are going to talk about nucleic acids in this third installment in our biomolecules section. Um, so let's get started. So for this section, by the end of it, you should be able to identify the building blocks of nucleic acids, explain the structural and functional characteristics of nucleic acids, describe the phosphodiester bond, compare and contrast deoxyribonucleotides and ribonucleotides, and explain the structural and functional characteristics of nucleic acids. Alrighty. So nucleic acids are DNA and RNA. These are macromolecules, right? They're polymers, which means that they're composed of many monomers. The two main ones that we talk about are DNA and RNA. So DNA is our double-stranded version and RNA is our single-stranded version. Now, the nucleic acid monomers, the individual components, are these guys here, right? So we've got these nucleotides, which has our nitrogenous base with a pentose sugar, which is a 5-carbon sugar, 1, 2, 3, 4, 5, and a phosphate group. Now it can have up to 3 phosphate groups, so it can have 1, 2, or 3. Now for our different nitrogenous bases, they can be our adenine, guanine, cytosine, or thymine in DNA. And in RNA, that thymine is subbed out for uracil. Now, likewise, our pentose sugar in DNA is deoxyribose, and our pentose sugar in RNA is ribose. And here we have an example of what all these things look like. So up here we have our nucleotide, and then down here we show ribose and deoxyribose. And if you look at it, deoxyribose has 1 less oxygen than ribose. So down here on this carbon, ribose has an OH group. Deoxyribose does not. So there's got 1 less oxygen, deoxy. Then this is what our different nitrogenous bases look like. We have cytosine, uracil, thymine, and then guanine and adenine. Now, as might make perfect sense, DNA or deoxyribonucleic acid is made of deoxyribonucleotides and therefore contains deoxyribose sugar. And again, I know I've said this multiple times, but I can't stress it enough. Our bases are adenine and thymine, guanine, and cytosine. And they always pair up in that fashion, adenine to thymine, or A to T, and guanine to cytosine, or C to G. Notice that the A and the T have 2 hydrogen bonds, and the G and the C have 3 hydrogen bonds. Now, our ribonucleotides are what make up RNA, and they contain the adenosine, the uridine, guanosine, and cytidine. And so for these, what we've got is, you notice that we have our ribose sugar, so it's got our 2 OH groups, so that indicates that it's RNA. And then we've got our 4 bases, right? So adenine, uracil, guanine, and cytosine, right? And then, of course, our phosphate. Now, when you polymerize these nitrogenous bases or these nucleotides, then you form a backbone. Those backbones are connected by what are called phosphodiester bonds, right? And so the upside about biochemists is that they tend to name things very straightforward. So phosphodiester, if you were to break that word down, it sounds like it has a phospho or a phosphate, and it has diester or 2 ester. So a phosphodiester bond is a phosphate connected to your 2 pentose sugars via ester bonds, right? So we have an ester bond from this C to O to P, and we've got an ester bond from this C to O to P. And then, of course, the P is our phosphate right here. Now, the formation of these phosphodiester bonds occurs in a similar fashion to when we make polysaccharides. We add on the additional nucleotide through a dehydration synthesis, and therefore H2O is released. And you could think about how would you break those bonds. It's going to break through a hydrolysis reaction again. Now, our nucleic acids have a couple of different functions. We're going to talk about a couple here. We'll talk about some more later on for RNA because they've got a bunch of different stuff. But DNA is going to be our genetic blueprint. It has all of our genes. RNA, one of the big things it does is it directs the production of proteins. There are a couple of different RNA types that are involved in that process, which we'll talk about later on. And then we have some specialized RNAs that can help catalyze reactions. Those are called ribozymes. And then, of course, there are special forms of nucleotides that can be used for energy, specifically ATP, which is adenosine triphosphate. So ATP is the primary energy currency of the cell. So we've got our adenine nitrogenous base connected to our ribose sugar, which we know because we have our two OHs here. And then we have our three phosphate groups. So that's what makes it triphosphate. These phosphate groups hold energy within them. So if you cleave off one of these phosphates, you're going to go to adenosine diphosphate. If you cleave off two, you'll go to adenosine monophosphate. So when you cleave off one or two of these, you end up releasing energy, and that's how ATP powers the cell. So at this point, you should be able to identify the different building blocks of nucleic acids, explain the structural and functional characteristics of nucleic acids, and describe phosphodiester bonds. You should be able to compare and contrast deoxyribonucleotides with ribonucleotides and explain the structural and functional characteristics of nucleic acids. With that, there is an objective check in D2L. Give it a whirl and see how you're doing on these objectives. See you soon.